Take a deep breath in.
Right? And just hold it for a second.
Now let it out.
Yeah.
So in the time it took you to do that, you just inhaled something invisible.
You really did. Dozens of them, probably.
Yeah, it's floating in the air of the room you're sitting in right now. I mean, a perfectly healthy person breathes in like dozens, maybe hundreds of these microscopic particles every single day without ever noticing.
Which is actually exactly how human biology is designed to handle this specific environmental presence. Our upper respiratory tracts and our immune cells, they're incredibly efficient at just sweeping these particles away.
Right. The invisible thing you just inhaled is a mold.
Yeah.
And, while your body might be neutralizing it effortlessly right now, a specific species of this exact same mold was named to the World Health Organization's first ever fungal priority pathogens list.
Yeah, back in 2022.
Exactly. And it was categorized as a critical priority. So they placed it right alongside, the most dangerous antibiotic-resistant bacteria on the planet, which is just wild to me.
It really presents this staggering biological contrast. I mean, you have an organism that is utterly harmless to the vast majority of the global population, right?
Yeah.
But then it is simultaneously capable of causing some of the most complex, lethal lung diseases known to modern medicine.
So to understand how an everyday mold manages to be, simultaneously harmless and completely lethal, we kind of have to look at what this organism actually is.
Right. The genus is called Aspergillus.
Aspergillus.
Yeah.
And surprisingly, the story of identifying this critical pathogen begins long before anyone even knew what a pathogen was, right?
Oh, yeah. It begins way back in 1729 with this Italian priest and botanist. His name was Pier Antonio Micheli.
Okay.
And he was working in Florence, just examining various environmental samples under an early, a relatively crude compound microscope.
So what did he see?
He observed a specific fungal structure. It's the microscopic stalk that basically bears the mold spores. Under his lens, it formed this radiating kind of spherical cluster of chains.
And to a Catholic priest in the 18th century, that microscopic shape looked exactly like an aspergillum.
Exactly.
The holy water sprinkler, like the metal ball on a stick that priests use to scatter water over a congregation during Mass.
Yeah, Micheli basically looks through his lens and sees a tool from his day job. And that is exactly how the entire genus got its name.
That is so funny. But back then, they didn't know it made people sick.
Right. Not at all. I mean, the historical context of that era is really essential here. In the 1700s, fungi were largely lumped in with plants.
Okay, yeah.
The scientific community had no concept that a mold could cause illness in a human being. Aspergillus was viewed purely as a saprophyte, an organism that just decomposes dead organic matter out in the environment.
So they just thought it was breaking down dead leaves and stuff.
Right. The connection between this mold and human disease wouldn't be firmly established until the whole germ theory era. Pasteur and Koch in the late 19th century.
But even after scientists realized it could cause disease, they still struggled to accurately identify it, didn't they?
Oh, massively. For most of the 20th century, mycologists categorized different Aspergillus species based entirely on their morphology.
Just how they look.
Exactly. They looked at the shape under a microscope, the color of the spores, how the colony grew in a petri dish, and they just sorted them into these visual families.
But I'm guessing looks can be deceiving.
Highly deceiving.
Yeah.
That purely morphological system was the global standard for decades. It allowed researchers to organize hundreds of isolates. But as we move into the genomic era in the 1990s and 2000s, DNA sequencing revealed a massive flaw in that system.
What happened?
Microbiologists realized they were dealing with what we now call cryptic species complexes.
Cryptic species complexes. So essentially, that's a group of organisms that are genetically distinct but look absolutely identical under a microscope.
Yes.
It's kind of like assuming two strangers on the street are biological siblings just because they happen to be wearing the exact same winter jacket.
That is a perfect analogy. The visual similarity masks a completely different genetic makeup.
And in a clinical setting, like a hospital lab relying just on microscopes, how much danger does that mistaken identity actually cause?
It can absolutely be fatal. Take Aspergillus fumigatus, which is the species responsible for the vast majority of human infections.
Good.
DNA sequencing revealed that fumigatus has numerous cryptic relatives. So, for example, a species called Aspergillus lentulus.
Lentulus.
Under a hospital laboratory's microscope, lentulus wears that exact same winter jacket. It looks completely identical to standard fumigatus.
So the lab tech looks at it and says, yep, that's standard fumigatus.
Mhm.
But those two strains behave very differently inside the patient.
They behave differently, and more importantly, they have completely different vulnerabilities. These genetically distinct cryptic relatives are often intrinsically resistant to our frontline antifungal drugs.
Oh, wow. So if the lab misidentifies the mold,
The physician might prescribe a standard antifungal, entirely unaware that the actual cryptic strain is immune to it.
and the treatment just fails.
The treatment fails, the infection spreads, and that's why taxonomists are actively having to rewrite the entire genus today to prevent these clinical blind spots.
Okay, so that explains the taxonomy side of things, but the physical organism itself presents another mystery to me. I mean, this mold is virtually everywhere, right?
Everywhere. Garden soil, compost, household dust.
Construction debris. So, to cause disease, it has to physically infiltrate our bodies.
Hmm.
How is it doing that so effectively?
Well, the infiltration really relies on two specific biological traits, and the first is spore size. Aspergillus reproduces by releasing asexual spores, which are known as conidia, right into the air.
By the millions, I assume.
Millions upon millions. And they are incredibly small. We're talking roughly 2 to 3.5 micrometers in diameter.
Just for some perspective on that for you listening, that is smaller than a single human red blood cell.
Yeah, it's tiny. And that microscopic scale allows them to bypass the body's initial defenses.
Because normally we filter dust out.
Right. When you inhale normal environmental dust, those larger particles get trapped by the mucus in your nose, or the little cilia lining your upper airway. But Aspergillus spores are so small, they ride the airflow directly past all those filters.
They just bypass security.
Exactly. They travel deep into the lungs until they reach the alveoli, which are the tiny air sacs where oxygen exchange actually occurs.
So they land in the deep air sacs. I mean, at that point, the immune system has to take over, right?
It does. In a healthy person, specialized immune cells called alveolar macrophages are constantly patrolling the alveoli.
Like little security guards.
Yeah, and the moment a spore lands, a macrophage effectively swallows it. It destroys it with enzymes and reactive oxygen species long before the spore ever gets a chance to germinate.
So the system works perfectly for most of us.
Flawlessly. But if the spore does manage to evade those macrophages, it relies on its second core trait, which is thermotolerance.
Heat resistance.
Right. The vast majority of environmental molds simply cannot survive at human body temperature, which is 37 degrees Celsius.
It's too hot for them.
Yeah, the environment inside the human body is just too warm for their cellular machinery to function, so they can't replicate. Aspergillus fumigatus, however, thrives at 37 degrees. In fact, it can tolerate temperatures all the way up to 50 degrees Celsius.
Which is fascinating because it's not like it evolved that heat resistance to attack humans, like a Goldilocks scenario. Most molds find us too hot, but this one finds us just right entirely by accident.
It really is an accident of evolution. It's an environmental decomposer. It actually evolved to survive in self-heating compost piles.
Oh, right. Because when dead vegetation breaks down in a compost heap, the microbes generate a massive amount of heat.
Exactly. And that compost connection really highlights the definition of an opportunistic pathogen. To survive its natural, highly competitive environment, this mold had to develop extreme thermotolerance.
And it just so happens that a survival trick for a steaming compost heap is exactly what lets it survive a human lung.
Precisely. It doesn't actively seek out humans. It just possesses the biological tools to capitalize if a human host's defenses drop.
So we have a microscopic spore that bypasses our airway filters, and it likes the heat of our lungs. But why do some people get horribly sick while others feel absolutely nothing?
That comes down to the host. Mycologists often visualize this spectrum of illness as a triangle of disease. The clinical outcome is dictated almost entirely by the host's underlying anatomy and the specific state of their immune system.
Okay, so let's break down this triangle.
Yeah.
The first point is allergic disease, right? Where the immune system doesn't fail, it actually overreacts.
Right. The classic presentation here is allergic bronchopulmonary aspergillosis, commonly referred to as ABPA.
BPA.
Yeah. ABPA typically affects individuals who already have underlying respiratory conditions, most commonly asthma or cystic fibrosis.
So what's actually happening in their lungs?
The mold spores essentially colonize the mucus in the patient's airways. Now, the fungus isn't actively invading the lung tissue itself, but its constant presence triggers the immune system to mount this massive chronic allergic response.
So the body is just fighting it constantly.
Yeah, constantly. And over time, that severe inflammation causes wheezing, mucus plugging, and eventually permanent structural damage to the airways.
This condition seems to create a really awkward dilemma for doctors.
Yeah.
I mean, if ABPA sits kind of on the fence between a pure infection and an allergy, how do doctors even know whether to treat the fungus or treat the allergic reaction?
That exact question has driven a really intense, albeit quiet, debate in the medical community for years.
I can imagine.
It comes down to nosology, which is the branch of medicine that deals with the classification of diseases. How you classify the disease dictates how you treat it.
Right.
For ABPA, this nosological debate has caused treatment guidelines to basically shift back and forth. Do you prescribe antifungal drugs to eradicate the mold, triggering the response? Or do you prescribe immune-modulating steroids to suppress the allergic inflammation that's actually causing the damage?
Which one do they usually do?
In clinical practice, it frequently requires a really delicate, highly individualized balancing act of both therapies.
Okay, so that's the allergy point of the triangle. The second point shifts away from the immune system and focuses on physical lung anatomy. This chronic disease.
Yes, also known as saprophytic disease. It occurs when a person has pre-existing structural damage in their lungs.
So, like, what kind of damage?
A classic example is a patient who previously survived tuberculosis.
Okay.
Long after the TB bacteria have been cleared, the disease leaves behind physical scars, or literally empty cavities in the lung tissue.
And the mold finds those cavities.
Exactly. When Aspergillus spores are inhaled, they can settle into these empty anatomical spaces.
It's like finding a perfect sheltered piece of real estate deep in the lung. But why can't the immune system just clear it out of that cavity?
It's due to a lack of vascularization. That scarred tissue surrounding the cavity has very poor blood supply.
Oh, I see.
Yeah, so the immune cells that circulate in the blood cannot effectively penetrate that space. Left undisturbed, the spores germinate and just grow into a dense, tangled mass of fungal threads.
What doctors call a fungus ball, right? Or an aspergilloma.
Yep. And over months or years, this can develop into chronic pulmonary aspergillosis. It slowly erodes the surrounding lung tissue and can cause severe, sometimes fatal bleeding.
That is terrifying. But the third point of the triangle is even worse, right?
Much worse. It's the most acute and lethal. Invasive disease or invasive pulmonary aspergillosis.
And this one primarily strikes the severely immunocompromised.
Right. We are talking about patients undergoing aggressive cancer chemotherapy, individuals who have recently had organ or stem cell transplants, or patients receiving massive doses of immunosuppressive steroids.
So their immune systems are basically offline.
Exactly. In those patients, the white blood cell count, specifically the neutrophils that patrol the deep lungs, they drop dangerously low.
So the security guards are just gone.
Gone. And without those neutrophils, the spores germinate entirely unchecked directly inside the healthy lung tissue. The resulting fungal threads, which are called hyphae, are highly invasive.
Meaning they don't just stay in the lungs?
Right. They penetrate through the lung tissue and literally invade the blood vessels. Once they breach the vascular system, fragments of the fungus can disseminate through the bloodstream to other organs.
Like the brain.
The brain, the kidneys, the skin. The mortality rate is exceptionally high.
I think we really need to clarify a common misconception here for everyone listening. Because when people hear the phrase deadly mold, the immediate image is the black mold growing on a damp bathroom wall or a leaky ceiling.
Right. Stachybotrys.
Yeah, but that visible indoor mold is an entirely different threat profile, isn't it?
Completely different. Visible household mold is primarily an allergy nuisance. It can certainly exacerbate asthma or cause respiratory irritation, but it is generally not a source of airborne invasive infection.
Okay.
The true lethal threat of Aspergillus comes from inhaling airborne spores from concentrated environmental sources like compost facilities, agricultural dust, or even contaminated hospital ventilation systems. And again, it only becomes an invasive threat when the host's immune defenses are severely compromised.
So really the host is destiny. I mean, the exact same microscopic spore inhaled by 3 different people can result in 0 symptoms, a chronic asthma flare-up, or a fatal brain infection.
Precisely.
So to see how this plays out beyond just the theory, let's look at some extreme real-world scenarios. The Toon Curse of 1973.
Oh, this is a fascinating case study.
It sounds like a movie.
Yeah.
It really is one of the clearest examples of what happens when a human encounters an overwhelming environmental source, right?
It is. In 1973, a team of researchers in Krakow, Poland, opened the tomb of King Casimir IV. Now, this tomb had been completely sealed for centuries.
And then they open it.
Right. And within weeks of entering the crypt, several members of the research team developed severe illnesses and died.
Which sounds exactly like the mythical curse of Tutankhamun's tomb.
Yeah.
But in Poland, they actually isolated the real culprit.
They did. A microbiologist investigated the air inside the crypt and successfully cultured a species called Aspergillus flavus.
So it was just waiting in there.
Basically, yeah. The prevailing scientific consensus is that a massive concentration of these spores had lain dormant in that sealed, undisturbed environment for hundreds of years.
So when they finally break the seal,
The air was agitated, and the researchers inhaled an incredibly high, concentrated dose. You see, even a relatively healthy immune system can be completely overwhelmed if the initial spore load is massive enough. It leads to the rapid onset of invasive disease.
okay, so that's an ancient tomb. You'd assume modern medicine, far removed from ancient crypts, would have this under control. But the COVID-19 pandemic actually created an unprecedented new landscape for this exact pathogen.
It really did. Intensive care physicians started documenting a sharp rise in a secondary infection called Kappa COVID-19-associated pulmonary aspergillosis.
So what was happening to these patients?
We observed patients who were critically ill on ventilators. They were successfully fighting off the viral pneumonia, only to subsequently develop and often succumb to invasive Aspergillus infections.
And this wasn't entirely new, right?
No, the medical community had documented a similar phenomenon previously with severe influenza cases, which is known as IAPA.
Considering the COVID and flu ICU cases, are our own modern medical interventions like ventilators and steroids inadvertently acting as an open door for this ancient mold?
Unfortunately, yes. A severe virus like COVID-19 physically damages the epithelial lining of the airways, which creates physical openings for the mold. But the clinical intervention is often what truly kicks the door wide open.
How so?
To prevent a patient's immune system from destroying their own lungs through inflammation during severe COVID, doctors administer high doses of life-saving corticosteroids.
Which calms the inflammation.
Right. But they also severely suppress the very neutrophils required to fight off an inhaled mold spore.
So we use ventilators to keep them breathing and steroids to calm their immune systems. Those save millions of lives, but they accidentally create the exact biological environment an opportunistic mold needs.
Exactly. Modern medicine is keeping far more vulnerable people alive today. Cancer patients, transplant recipients, people relying on biologics. By extending their lives, we are simultaneously expanding the population at risk for invasive fungal diseases.
But I want to pivot for a second because not all Aspergillus species are killers, right? The industrial side of this is actually wild.
It is. The genus diversity is remarkable.
Because while fumigatus is causing havoc in ICUs, another species in the exact same genus, Aspergillus niger, is mass manufacturing stuff for our food supply.
Yes, A. niger is responsible for manufacturing citric acid.
Since 1919, I read that a chemist named James Curry did the research, Pfizer commercialized it, and now this mold is fermented in massive vats to produce the sour flavor in almost every soda and candy on Earth.
It's incredible. And you also have species like Aspergillus clavatus, which causes an allergic condition called malt worker's lung in brewery workers who are exposed to germinating barley.
So I guess I'm never looking at soda or ancient tombs the same way again.
Probably a safe bet. But for the clinical community, the urgent focus remains strictly on the pathogenic species, and how we actually fight back.
Right, and the fight against invasive aspergillosis has evolved a lot. For decades, the frontline defense was a drug called amphotericin B.
Yes, amphotericin B. It was effective at destroying fungal cells, but its mechanism of action made it notoriously brutal on the human body. The medical community literally nicknamed it amphoterrible.
Amphoterrible. Why was it so bad?
Well, it works by binding to ergosterol, which is a component of the fungal cell membrane, and it basically tears holes in it.
Sounds effective.
It is, but the problem is that ergosterol is structurally very similar to human cholesterol.
Uh-oh.
Yeah. The drug couldn't always distinguish between the two, so that led to severe collateral damage, most notably extreme kidney toxicity, and just intense infusion reactions in the patient.
So they were basically poisoning the patient to kill the mold. But the treatment landscape completely shifted in 2002, right?
It did, driven by a landmark clinical trial led by researcher Raoul Herbrecht.
And the Herbrecht trial fundamentally altered global treatment guidelines almost overnight. They tested the toxic amphotericin B against a newer class of drug, a triazole antifungal called voriconazole.
And the clinical data was undeniable. Voriconazole demonstrated significantly better patient survival rates and caused just a fraction of the toxic side effects.
How does it work differently?
Triazole drugs target the enzymes that synthesize the fungal cell membrane rather than tearing holes in it directly, which makes them far safer for human tissue. Today, azoles like voriconazole remain the primary standard of care.
But having a safer drug only works if you actually know what you're treating, and diagnosing invasive aspergillosis is famously difficult.
Incredibly difficult. A standard blood culture often fails because the fungus primarily invades deep lung tissue. It doesn't tend to circulate freely in the bloodstream until the very, very late stages of the disease.
So they can't just draw blood, and I'm guessing a lung biopsy is too dangerous.
Way too dangerous for an ICU patient who is already unstable. Consequently, clinicians heavily rely on non-invasive biomarker tests, and the most common one is the galactomannan test.
Galactomannan? What is that?
It's a specific complex sugar molecule that makes up the cell wall of Aspergillus. As the fungus grows in the lung, it sheds these sugar molecules into the bloodstream, which the test can then detect.
But there's a major debate around this test, entirely because of a bizarre cross-reactivity issue with certain antibiotics, right?
Yes. Patients receiving certain common intravenous antibiotics, specifically piperacillin and tazobactam, were routinely testing positive for the fungus even when they absolutely didn't have it.
Wait, so the antibiotic itself triggers a false positive for the mold. How does that even happen?
It's fascinating. Many of our standard antibiotics, including piperacillin, are fundamentally derived from penicillium molds.
Like penicillin?
Exactly. Penicillium and Aspergillus are closely related fungal genera, and they share almost identical galactomannan antigens in their cell walls. Right. So the manufacturing process for the antibiotic doesn't completely filter out those leftover fungal sugar molecules. When the antibiotic is infused into the patient, the test detects those harmless sugars and falsely signals an active Aspergillus infection.
That sounds like a nightmare for infectious disease specialists.
It is. It forces them to constantly weigh the results of that test against DNA testing and clinical CT scans just to confirm a diagnosis.
Wow, so the diagnostic hurdles are complex, but the biggest controversy right now actually involves the treatment itself. The azole drugs, like voriconazole, are starting to fail.
They are. Aspergillus fumigatus is developing widespread azoles resistance, which is exactly what prompted that WHO critical priority warning we talked about earlier.
And initially, people assumed this resistance was happening inside the hospitals, right? Like, long-term use of the drug in chronic patients was forcing the fungus to mutate.
Right, and while that does happen, the broader medical community is now focusing intensely on what's called the One Health hypothesis.
The One Health hypothesis, basically arguing that human medicine and agricultural practices are deeply interconnected.
Exactly. And it points the finger directly at how we protect our crops. Global agriculture relies heavily on massive quantities of triazole fungicides.
To prevent mold on crops and timber.
Yeah, on commercial flower bulbs, yeah. But on a molecular level, these agricultural fungicides are nearly identical to the life-saving azole drugs used in human hospitals.
So if farm fungicides are basically the same drug and we are just continuously saturating the soil with them,
Yeah.
the Aspergillus living in the dirt is forced to adapt.
Right. It evolves genetic resistance to the agricultural triazoles just to survive out in the fields.
And once it evolves that resistance in the field,
Its spores are carried by the wind. So picture an immunocompromised patient in a completely different environment. They inhale one of those wind-borne spores. They develop an invasive infection that is already fully resistant to our clinical azole drugs.
even though that patient has never taken an antifungal in their entire life. That is like a horror movie, where the monster becomes immune to your weapons before you even meet it. How do policymakers even begin to balance crop protection with saving ICU patients?
It's an incredibly difficult policy dilemma, particularly in places like Europe, where this debate is really active right now. Balancing agricultural yields with critical human pharmacology, it requires an unprecedented level of global coordination.
Which is why the WHO is sounding the alarm.
Exactly. For decades, the focus has been entirely on antibiotic-resistant bacteria, but environmental fungal resistance is proving to be a parallel, deeply entrenched threat.
The journey of understanding this organism really is incredible. I mean, we go from an 18th century botanist mistaking a microscopic stalk for a holy water sprinkler, to recognizing it as a heat-loving opportunist exploiting modern medical interventions, and now it's tangling with global agriculture.
It really is. And the ultimate takeaway here is that the presence of the fungus is only half of the equation. Aspergillus spores are a permanent, ubiquitous fixture of the air we breathe. It is the intricate, highly individualized interplay with human biology that dictates whether the spore is silently destroyed, triggers an allergic cascade, or initiates a lethal invasion.
We inhale it constantly, but the body itself is the final deciding battleground. There is, however, one final lingering thought here that goes far beyond the hospital walls.
The environmental catalyst for its survival.
Exactly. The foundational reason Aspergillus fumigatus can infect us at all is its thermotolerance. It evolved to survive the intense 50-degree heat of a rotting compost pile. But as we look toward the future, the global environment itself is fundamentally shifting.
Right.
So for everyone listening to this deep dive, think about this. What happens to the vast, undiscovered remainder of the fungal kingdom as climate change continues to warm the Earth?
It's a sobering thought.
Could rising global temperatures effectively act as a planetary-scale compost heap, applying evolutionary pressure that basically trains entirely new, previously harmless species of environmental molds to survive the heat of the human body?
It is a profound, entirely plausible question that the scientific community is really just beginning to model.
Definitely something to consider the next time you take a deep breath.